We're very pleased to welcome you to this RBMO webinar, the 16th edition of our quarterly webinars, which we have been running in collaboration with i3. We're very happy to be back and for this special session to showcase the winners of our annual Robert Edwards Prize Paper Award. This Prize Paper Award is the best papers published in RBMO in 2025, as Mina was saying before. And these prizes are based on the number of citations they get, the number of downloads, and the votes from editors and section editors. We will introduce our first speaker short, but please let me first introduce my co-host for today. We'll keep our introductions short tonight, but you can see all our extended bios on the i3 website, ivfmeeting.com. And I'm absolutely delighted to be joined by my co-host and editor at RBMO, Dr. Mina Alikani. Dr. Mina Alikani is a clinical consultant, embryologist, and board-certified high-complexity lab director with extensive experience in IVF lab leadership and quality management. She is the past president of Alpha Scientists in Reproductive Medicine and one of our senior editor team at Reproductive Biomedicine Online. She has authored more than 80 scientific publications, is an internationally recognized speaker, and also a consultant for conceivable life sciences.
Thank you so much, Juan. It's a great pleasure to be here with you to host this special RBMO session. And now let me tell our audience a little bit about you. Not that you need an introduction. I think everyone here knows you. Juan Garcia-Velasco is the Chief Scientific Officer of EVRMA Global, Director of EVRMA Madrid, and Professor of Obstetrics and Gynecology at Rey Juan Carlos University. He's a world-renowned expert in IVF, as you know, our audience would know, and has published over 200 scientific papers and serves as co-editor-in-chief of RBMO alongside Nick Macklin. Now, as we move to our presentations, as always, we encourage you to engage with the speakers and ask questions. But please remember to put your questions in the Q&A section and not in the chat section where they might be missed. So now, Juan will introduce our first speaker and the prize winner, the RGE prize winner.
Thank you, Mina. And exactly, without further ado, it is my pleasure to introduce our first speaker tonight, Dr. Gerardo Mendizabal. He's a researcher and inventor specializing in IVF automation, robotics, computer vision, and artificial intelligence. His work focuses on developing innovative technologies that improve precision, efficiency, and clinical outcomes in assisted reproduction, helping drive the future of reproductive medicine. He was the prize paper of 2025 in RBMO. So, Gerardo, please.
Thank you very much for the introduction, and thank you all for being here. I am here today to share the results of a project that we have been working for some time. This is building and testing a system that uses artificial intelligence and automation to perform ICSI remotely. So over the next 20 minutes or so, I will walk you through why we believe this is important, why it matters, how the system is built, the AI behind it, and the preclinical and clinical results we have obtained so far. So first, my conflicts of interest. I'm the VP of Exploration at Conceivable Life Sciences. I'm also a research professor at the University of Guadalajara in Mexico, and I am a shareholder in both Conceivable and IVF 2.0. Intracytoplasmatic sperm injection, ICSI, you all know this, was introduced in the early 90s, originally as a solution for severe male factor infertility. It works by injecting a single sperm directly into the oocyte, bypassing the natural barriers to fertilization. Today, ICSI is a method of choice for insemination during IVF, and it is used even when male factor is not the issue. To give a sense of scale, according to the European IVF Monitoring Consortium, 73% of all reported treatment cycles in 2019 were performed using ICSI. So this procedure is nowadays kind of in the center of modern assisted reproduction. Performing ICSI requires an inverted microscope, a set of specialized micromanipulator tools. With this, the embryologist selects a single sperm, immobilize it, and then microinject it into the oocytes. Each of these steps demand extraordinary precision of steady, fine motor control at the level of a single cell. Therefore, mastering ICSI takes far more than understanding the theory. It also requires hands-on practice. It needs to be performed repeatedly under the supervision of an experienced professional. And depending on the trainee's prior background and then their natural aptitude for the work, that training can take a considerable amount of time. So, because there are many ways to perform ICSI, there is not a single rule how to do it. Every embryologist ends up developing their own unique approach, their own technique, their own habits, their own subtle decisions. So these variations can lead to real inconsistencies in the quality and success rate of the procedure. And that is true even within a single clinic, between people working side by side. So this is exactly why standardization matters in IVF. So our goal should be that every patient can receive the same high quality of treatment, regardless of which clinic they walk into, and regardless of who happens to be at the time in the microscope, or if anything happens to them, right, during the day, if they tire, I don't know, many things can happen that can change the performance. So how we're tackling this problem? Well, we can find inspiration for a solution by looking at surgery more broadly. So probably many of you are familiar with this robot. This is a robotic surgical system named the Da Vinci. And I think this is the most familiar example where this system was designed to extend the capabilities of the surgeon, particularly in procedures that demand very high precision. So a surgical robot improves precision, flexibility, and control during the operation. And in doing so, it also minimizes risk. But there is also a second advantage. These systems can allow the operation to perform, the operator, to perform the procedure remotely from a different location entirely. So that means that the specialized surgical expertise can be brought to isolated areas, directly addressing healthcare disparities and expanding access to advanced care for patients who otherwise wouldn't have it. So following that exact idea, we have developed a single-cell robotic surgery system that is capable of performing the full ICSI protocols autonomously under the remote supervision of an embryologist. The objective is that this system can deliver more consistent results. One, that reduce the variability in how the procedure is executed, that minimize the risk of human error, and in the future, that can help reducing the cost and improve the overall success rate. So in short, we're trying to take the standardization that surgical robots brought to the operating room, and we're trying to apply it at the scale of a single cell for IVF treatments. So at its core, the system combines optical elements and high-precision robotic micromanipulators, coupled with intelligent visual servo controls that are powered by 5 specialized AI models. It's important to mention that we didn't build this from scratch. We built this system around a commercially available Olympus IX81 microscope. Many of you are maybe familiar with this one. But we fitted a digital controllable stage and objective turrets together with the digital Epenor micromanipulators. So on top of that, the system also incorporates a Hamilton laser, a DTS laser, which we use both to immobilize the sperm, the selected sperm, and also to thin the zona pellucida before the needle penetrated in such a way that we can reduce the amount of stress that we put to the egg when penetrated. Also, we also include a piezo system, which is used to open, to break the olema prior to the depositing of the sperm. So we carefully integrated to this platform, and we built largely on hardware that the field already knows and trusts. So as I say, We're not inventing this from scratch. We're using things that already have been used many years in the IVF industry. So the whole system is operated through a set of on-screen digital controls. From this, the operator can modify the configuration of the optics, the position of the stage, the position of the micro tools. Everything that the operator will normally do by hand at the microscope now is available through this digital interface, which of course is precisely what makes remote operation possible. So, but it's not only these tools that we have on the screen and that can perform the digital control. We want most of the things to happen automatically, and for that we are using 5 deep learning models that handle computer vision tasks. That means identifying structures of interest within the microscopic images. So each one of these AIs have a specific job. The first model is meant to detect and track sperm in PVP, allowing the system to assess the motility and morphology patterns in real time. This is pretty much seed that we developed when I was in IVF 2.0 is exactly the same algorithm. The difference is that once this system select the best sperm, it automatically bring the sperm to the center where we have the laser, and it allows us to do a single shot that immobilizes the sperm, the selected sperm. So the other two AIs that we have here are object detectors. So they find bounding boxes around the tip of the holding a pipette and the tip of the ICSI needle, the egg and the egg at different magnifications, and the sperm inside the needle, as well as the meniscus. So we use that for driving everything, right? Where to position the needle, where to position the holding pipette, and the amount of aspiration that we need. Also, the final 2 models perform semantic segmentation of the oocytes and the sperm cells, and this allows us to identify the different structures of the gametes. And those structures are what the system used to make decisions to the procedure. Altogether, these 5 models give the system both the information it needs to act by its own. So all of the perception fits into the system's automated routines. In total, there are 15 of them, 7 dedicated to the sperm side of the procedure and 8 to the egg side. Now, I want to emphasize this point. The operator is always in control. He or she has the capacity to interrupt any of these routines if necessary. At that point, using the digital controls, the operator can make whatever adjustment the situation requires, and then resume automatic steps from there. So this is autonomous, but it is supervised autonomy. The human expert is always to be able to step in. So if we look one level deeper, the sperm routines and cell break down further into 23 automated subcommands. Each of these handles a small, well-defined part of the task. And on the X side, the routines break down into 34 subcommands. To many those 15 high-level routines, there are a great deal of fine-grained, careful sequenced automation doing precise work. So rather than just describe the system and then automated protocol, let me show you. This is a video that depicts an example of the robotic ICSI procedure from start to finish. If you go to the publication, there is a link that you can access and have a version of this video with more explanation. This one is just to show you how the seed selects the sperm and immobilizes it. Then here's to see how the system positions the needle on the tail of the sperm. It absorbs it, the AI finds it inside the needle and positions it to a stable position within the needle. Then it goes to the egg where there's an egg, finds the egg, position it under low magnification, change to high magnification, bring this holding the pipette, perform the automatic focusing. This is a step where we do a shot in the zona pellucida to thin it. Unfortunately, the quality of the video maybe you cannot see very clear, but there's some thinning there. Then the sperm is positioned next to the tip of the needle, and then the system performs penetration. Do the piezo pulse you will see there to break the lemma that happens there, and then finally the sperm is deposited into the cytoplasm. You can see that every time the system is going to do something, there's this black square there. So the user can always cancel if the user determines that some digital interventions has to be made. So there is always a countdown, 3, 2, 1, and the user can just interrupt if necessary. So of course, before anything approaches a clinic, the central question is if this thing is safe. So we performed preclinical validation studies to assess the safety of the laser mobilization of sperm. For this, we use a mouse model. The results were very encouraging. We achieved approximately 99% of normal fertilization rate, 80% of blastocyst formation rate, and about 40% live birth rate. So critically, we don't find any significant differences when compared with the manual control arm, in which the sperm were mobilized mechanically. In other words, the laser-based approach performed on pair with the established manual technique. In addition to that, we performed post-injection survival tests, this time on both hamster and human eggs. Here, the survival rate was about 94% in the hamster eggs and 93% in the human eggs. The results gave us confidence that the procedure itself was not compromising the viability of the oocytes. So having established safety, we moved to the clinic. We used the system to perform a total of 4 clinical cases, and each of these, both of the supervision and the operation of the system were carried out remotely. This was a real test for the central premise, right? Can we do this with an expert somewhere else apart from the clinic? And we did this with somebody from New York, supervised from New York, and we even did it transatlantic from Spain. So for the specific case that we report in the paper, these are the results. The system completed the procedure with digital interventions. There was quite a lot of intervention, I would say, like at least half, 57 out of the 115. And this is because sometimes the autofocus was not good, or maybe we need to bring the needle closer, things like that. Normally, as I explained before, the operator just refine that procedure and then hit continue, and then the other steps happen automatically. So because of that, the procedure averaged around 10 minutes, roughly 4 minutes to process sperm, 1.5 minutes for egg preparation, and 4.5 minutes to deposit the sperm. Of course, these times are not ideal, and they are longer than the manual ICSI takes today. But it's important to notice that this can be explained because also in many parts we're having these opportunities for the user to stop the procedure. So that we have to see what's the plan of the automated system, and then we can cancel if we think that is not a good is not something that we don't want to happen. But anyway, this was an early case, and bringing those times down is exactly what the next generation of the system is built to address. So looking at the border clinical pilot trial, 22 oocytes were assigned to remote ICSI. With this, we achieved 95% survival, 81% fertilization, and 65% blastocyst formation. And the key point is that these results were comparable to manually performing ICSI controls. So we did per sibling oocytes, which makes the comparison especially meaningful. So performing remotely and largely autonomously, the system matches conventional manual ICSI. So we are now currently working on the next generation of the system, and the focus there is scalability. So we're running a new study, and in this one, we're already researching, we're already reaching lower procedure times. Still some work to do, but this is an important step if this is going to be deployed widely and practically in the future. And one thing that I want to highlight is that this system, we have an inverted scope, OEM's inverted scope, and we also adding 2 temporary incubators. So the idea is to reduce the amount of time that the eggs are away from ideal conditions. So these temporary incubators is not only temperature, it's also gassed. So the idea is just to take the egg from the temporary incubators, put it on the ICSI microscope, do the injection, and right away just put it into the other incubator. So we minimize the time that is outside. This is how the system looks like. So bringing all this together, we are also building not only for ICSI, we're having other systems that we call Aura, and this is we have one for finding, washing, one for spread preparation, one that makes dishes. That's the one that many people like it. One for culturing, and also one for wheat. So, well, bringing this together, we have shown that the remote autonomy is feasible. We establish a safety in preclinical across mouse and hamster and in humans. And our clinical pilot, the results were comparable to manual ICSI. So next generation is going to be faster and easier to scale. So that's all I have for today. Thank you very much for your attention.
Thank you, Dr. Mendizabal. It's a pleasure to listen to this very exciting work, and congratulations for the award. We will have time for questions later, but just very briefly, let me ask you one simple question. Well, maybe not so simple, but if you had to choose just one advantage of this way of doing ICSI, whether it's reducing variability or reducing human workforce or I don't know, increasing results, what do you think is the main advantage of a system like this versus what we have today?
So I would say that maybe not now, because we're still working with this system and we're improving it, but over time we're going to achieve reduction in variability. So we're going to be very consistent within the results that we have in the injections. The aim is to lower the regeneration rates and increase over time, as I say, the blastocyst, the usable blastocyst formation rate. So since every injection is going to be standardized, I think that's very possible. So I will choose that one.
Okay, thank you very much. So we can continue then. Maybe Mina, yes.
Great. Thank you so much. Sorry, I'm losing my voice. It's bad timing. So let me introduce the runner-up prize paper, which is by Dr. Stephen Vasilescu and colleagues, and it will be presented by Stephen along with Dale Goss. So a little bit of an introduction. Stephen is a biomedical engineer and entrepreneur and founder and CEO of Neogenics Biosciences. He leads the development of innovative microfluidic-based and AI-enabled technologies for sperm selection and analysis, including the Sperm Guide, biomimetic, and you need to define that term, by the way, Stephen, I hope you will, sperm sorter. Joining him is Dale Goss. It's a pleasure to have him on this platform again. I have actually hosted another session where Dale presented. He's a great speaker. He's the Chief Scientific Officer of Neogenics Biosciences, who leads the development of microfluidic and AI-driven technologies, again, for sperm isolation and computer vision-based sperm detection in IVF. He's an embryologist and a PhD candidate at the University of Technology in Sydney. When do you expect to finish your PhD there?
This year.
Almost there.
Yeah.
Thank you. Okay, Stephen, please, you can take the floor.
Thank you, Mina. Everyone can see that? Yes?
Yes.
Fantastic.
Yes.
Awesome. So thank you for the introduction. It's great to be here, and thank you again to RBMO and everyone that's been supporting us. So over the next few minutes, I don't want to just show you the results that we achieved in our paper. We thought I could take you behind it and give a little bit of background on the development story, and then I'll hand over to Dale to tackle the results and the clinical results that we got from our paper regarding sperm selection. So I'll take it away. So a couple of quick disclosures. As mentioned, we have a startup and obviously I'm a shareholder in that. So let's start with the problem, and this goes all the way back to my PhD. Density gradient centrifugation is still the workhorse in most labs for sperm selection, or maybe you're doing a swim up. But we've known for a long time that the way we prepare sperm can damage these very cells that we're trying to select. Centrifugation generates shear stress and reactive oxygen species that drive strand breaks, and they can oxidize, and the end result is DNA fragmentation in the sperm. And it's more than just that. The process itself is operationally inconsistent and unstandardized across a lot of labs. So we thought we'd tackle this problem. And as you do, you go to see what is out there. And there's a rich body of work that looks at sperm selection mechanisms, or the mechanisms that they encounter in the female reproductive tract. And these are just a few. You've got thermotaxis, chemotaxis, rheotaxis. There's a lot of genuinely interesting ideas that we can tap into for selection, but where a lot of work can come undone, at least as far as translation goes, and that which is but drives everything that we do, is that they might not at the academic level have the sophistication or the simplicity to translate cleanly into an IVF lab. Can an embryologist run these technologies in a very busy lab? And I think most academic works, when you look at sperm selection, sit in the kind of top left here, where they have maybe a quite high degree of biological sophistication or technical sophistication, but they maybe don't translate extremely well. They take too long, there's too many steps involved, and you're not always considering the user that you have in mind, the embryologists that are using these devices. Really, you want to be sitting somewhere up here where you've got really good operational feasibility, but you've got a level of sophistication that means you're selecting sperm effectively. So what's sitting in the benches IVF labs right now is we have some commercial microfluidic devices, things like Zymont, Harvester, Lens Hook, and so on, that they are low impact and they reduce centrifugation, and there's reasonable clinical evidence behind them. But if we get technical, a lot of these are membrane-based devices with a plastic housing, and sperm can migrate through a microporous membrane under their own motility, and that's a meaningful improvement over DGC or swim up, but it's not the same as a true microfluidic channel exploiting some of these biomimetic principles that I'm going to mention. So we thought we could take it a step further. We can build something that leverages sperm motility in a more meaningful way. And we started looking more carefully at thigmotaxis, so the tendency of sperm, motile sperm, to follow walls and corners. And there's a lot of good work that has been done by Reza Nosrati, for instance, looking at how sperm turn corners, follow boundaries, and you can exploit that to mediate sperm selection. So using this passive hydrodynamic effect, that's exactly how sperm navigate naturally. And we thought, well, let's see if we can, how detailed we can get into that, and what we can do with our microfabrication techniques to achieve meaningful selection. So I thought I'd give a little bit of background of how we started iterating, how we started building. So we leveraged 3D printing. Traditional microfluidics and standard photolithography can take weeks to iterate your designs on. It's a very slow turnaround to test a device, and maybe it doesn't work, and then you have to go back to the drawing board and do the whole lithography process, which can be very expensive. What we developed is we started using these high precision 3D printers specifically for microfluidics, and we turned that few weeks turnaround time into a single day. So you can 3D model, you can CAD your design, 3D print a mold, cast it, test it all within the same day, and then repeat it the following day. So you can iterate very, very quickly, and that really unlocked the ability for us to test lots of different designs, lots of different geometries within a very short period of time, and quite relatively cheaply as well. So this is an example of some of the very, very first iterations of molds. You see you've got molds up the top here, and these are the polymer casts that come off that you peel off the top of those. And what we also realized we can start doing that we couldn't do before with these printers is we could play in 3 dimensions, right? We're not looking at 2D geometries anymore. We start looking at 3D geometries, and that opened up a whole new world of possibilities to mediate sperm selection, and get sperm where we want them. So we created new layers for sperm migration, tight junctions that sperm can interact with, boundaries to separate the semen and the buffer so samples stay very clean, and Dale talk a little bit more about that. Areas where we can trap sperm so that they don't swim back from the way they came, and we keep them where we want them. So there's a lot in a relatively simple chip that's very simple to operate. There's a lot of mechanisms that you don't see within the device that have been slowly optimized over time. And our first iteration, you can see some of the channel geometries here. We ended up with multiple levels, floors, if you like, for sperm to swim down to increase yield and to see how they can perform in different stages. Now we also have 3D geometries of sperm swim up and down to mediate their selection. And we started seeing quite good reductions in DNA fragmentation compared to conventional methods, which was, density gradient or swim up in this case. It's a bit of a roller coaster depending on who's operating, who's performing the test. But a printed prototype is not a product, and this is part of the journey I think that gets skipped all too often when you're 3D printing devices and they look fantastic, and it's not necessarily scalable. So the real test is whether you can make one elegant device, isn't whether you can make one elegant device, whether you can make tens or hundreds of thousands of them identically with reproducible results. And where we ended up having a lot of conversations with embryologists like Dale at the very, very beginning, people telling us, keep it simple, don't make the device too operationally complex for this particular tasks, pays dividends when you get to scale manufacturing, because the complexity compounds when you need to do it at scale. So we started generating some aluminium molds and then medical grade plastics off the top of that. You can see some of the units that we've developed that we're testing now. The fluid dynamics of those devices, the biocompatibility, which is looking quite good. So there's still a lot of work to be done. Dale will touch on some of that. But once we started looking at these components, we thought, okay, awesome. Now we can move into testing into these into clinics with these more robust design features, keeping manufacturability in mind. I'll pass it on to Dale now to chat more about the clinical results and what we've achieved in the paper. Thank you. Dale, go ahead.
Awesome. Thanks very much, Steve. So yeah, I get the pleasure of speaking about our paper in RBMO. And this was a project led by Steve and a team spanning across the University of Technology, Sydney, Monash Uni in Melbourne, and Melbourne IVF also in Melbourne. So these are my disclosures. I'm the Chief Scientific Officer of Neogenics. So Steve's given you a bit of a good background on the problem, but so here is the prototype of the solution that we wanted to present. So obviously we wanted to provide a gentler alternative to the centrifugation-based methods, so DGC in particular, and we wanted to use these biomimetic principles. So biomimetics is basically trying to mimic nature and the mechanisms that nature is using in the female reproductive tract in this specific case. So the device uses it's a passive system where motile sperm are actually self-selecting during the incubation process, and it uses this mechanism that Steve mentioned called thigmotaxis. So the device is fairly simple to use. It takes 1 ml of semen. The incubation process for the study was 15 minutes, and it was done at 37 degrees. So you basically then wait the 15 minutes and then perform an aspiration from the central outlet with media and your selected sperm. So it's 4 steps, like I said, fairly simple operation, and it's using common consumables that are found in the IVF lab, so general sperm buffers, pipettes, and syringes. So here you can see on the left is a micrograph of the semen on the left, where the sperm, or it's raw semen, and the sperm are moving into the channels and traversing them using that boundary following behavior. So they sort through a sort of microfluidic obstacle course to get to the outlet. And then, of course, only the highly motile sperm are able to traverse that those channels and actually get to that point. So you can see here the difference between the raw sample on the left and then the selected sperm on the right. So highly motile, progressively motile sperm. These lines are actually showing the outlet central collection zone where there's a step gradient so that it retains the sperm in that area so they can't swim out again. So to test the device against the DGC, so what was actually presented in the paper, we built the study around 2 phases, so 61 men in total, and it was based at 2 different sites. So phase 1 was a proof of concept based at Monash University, which was a university research lab. And then phase 2 was done at a diagnostic andrology lab in an IVF clinic. So the samples were assessed like a split sample comparison, where we compared the raw DGC and microfluidics for a few different outcomes. And those outcomes were sperm DNA fragmentation. We used sperm chromatin dispersion assays, so SCD, and then we assessed the progressive motility and concentration of the outputs. So I'll start with the proof of concept, where we used student donors to provide samples. So there were 21 donors in total. So most were fairly good samples to begin with. When we look at the DNA fragmentation assessment, so there's clear steps down from the raw to the density gradient to the microfluidic device, where the DGC was showing around half the DFI of the raw, which was around 5%. And then the mean DNA fragmentation for the microfluidic device went below 1%. So it was about 0.9%. So the average reduction was 92% for the microfluidic device and around 60% for the density gradient. When we looked at the progressive motility, we were getting consistently higher progressive motility in the microfluidic device group versus the DGC. And then obviously they're both significantly better than the raw sample. And then finally, when we looked at the concentration, so with this stringent sperm selection, there is a price to it, and that's the yield limitation, where we showed a mean of around 3.5 million per mil in the microfluidic device versus a mean of around 70 million for DGC. But you can see there's quite a variation in the raw starting samples too. So we progressed to the more a clinically relevant population of men attending the diagnostic andrology clinic within that IVF unit, Melbourne IVF. We observed similar pattern with this cohort. So in the 40 patients that attended, we had 33 that were included in the DFI assessment. So the DFI showed the same pattern as a proof of concept study. So a clear step down from raw to DGC to the microfluidic device. Where the DGC, where the microfluidic device had an average of around 2% DFI and around 6.5% for DGC. So much greater variability with the DGC, you can see with in this middle column here. When we looked at the progressive motility, interestingly, the motility was almost the same or very similar for both DGC and microfluidic selection, but both considerable improvements over the raw sample. And then finally, we can see the similar cost to the concentration, but in this case, with the lower starting concentrations for the raw, the difference between the microfluidic device and the DGC is much less, but still 4 million for the microfluidic device and then 25 million for DGC. So what does this all actually mean for how we might use the device in the clinic? So the bottom line is that the microfluidic device reduces DFI significantly more than DGC, as we showed, and it enriches a population of highly motile, like progressively motile sperm. So it does this by allowing the motile sperm in the device to self-select, and that's a method the method it's using is what the female reproductive tract already uses in natural reproduction. So we don't need centrifugation, we don't need chemicals, it's just using geometry to get those sperm out. The limitations, like I mentioned, modest total yield. And then for high responders, it may limit using conventional IVF, but we do hypothesize that this form of selection is leading to an enriched pool of sperm, which we could hope would require fewer of these sperm per oocyte during droplet IVF. But obviously, that's something that needs to be validated. But if we could use fewer sperm in droplet IVF, we'd also have less reactive oxygen species that the oocytes will be exposed to in the dish over the fertilization process too. And then how does this integrate into the clinic? So the device is clearly nicely suited for ICSI, and it may be indicated for men with high DFI, so which a lot of these devices are aimed at anyway. So chip-based methods are also fairly simple and reproducible, and they require very little equipment that the clinic has to set up. And this simplicity of our device, I think, fixes the failure of earlier channel-based methods, which can be complex and temperamental and can fail based on their sensitivity. So everything I've shown so far is the published research device. Steve alluded a little bit to the other device in his talk, but this is the kind of brief look at where the technology is headed next. So we've optimized the 3D geometries for the sperm migration, better sperm migration, and then also stability. And this is all done using medical grade plastics. And we've also spent a lot of time trying to ensure the output purity of the device. So in these new iterations, like we're finding that a common issue with current filter-based methods on the market, ones that are commercially available, is that they cause semen contamination in the output. So we've shown this, you can see here on the right, with the dye as well as using lateral flow assays. We've shown that dyeing the semen, you're getting an output in these filter-based devices of dye. And then when we do the lateral flow assays, which detects with quite high sensitivity sperm or seminal plasma proteins, specifically seminogelin, we're finding that we're consistently finding those proteins in the output. And then we believe that this provides a risk that channel-based methods actually avoid pretty well. So yeah, and we're also working on prospective studies on more clinically relevant DFI levels to assess how the devices are tackling these samples, and then of course moving on to the clinical outcomes that we want to see in the IVF lab. Yeah, so none of this happens alone. So I just want to extend a big thank you to the teams involved, of course the Neogenics team, and then the team from Monash University, Dr. Reza Nosrati and Dr. Fab Horta, and then of course the amazing team at Melbourne IVF that have helped so much with the study. You guys did a very good job. Thank you very much. I'll stop there, and of course we'll be happy to take questions.
Thank you very much, Dale and Stephen, for this nice presentation, and congratulations again on being the runner-up, winning the prize for runner-up paper. We are going to open Q&A in a minute. I just wanted to ask you a quick question, whether the quality of the sample at the start is a limitation of successful processing in your device.
I think that's a good question for you, Dale, because you've tested the lower limits of the device.
Yeah. So I mean, it'll, to an extent, it will be. The variation obviously depends largely on the total motile concentration, but we tested it to pretty low levels. I think we've gone as low as 0.1 million per mil motile concentration, and we're still able to get sperm at the end. It's just how much does the embryologist want to have to select from? So you're going to be reducing the population down considerably. We just want to make it as easy for the embryologist as possible to have a population of sperm to choose from during ICSI. But we could go as low as 0.1 starting, and you can still be able to have an abundance of sperm to start with.
Right. Thank you so much. I'm going to invite Gerardo and Juan to join us so we can start the Q&A session. And of course, I encourage the audience to participate by posing questions to this panel. Juan, would you like to continue with the question?
Yes, we have like 15 minutes and we have a lot of questions in the Q&A. Happy to receive more of this, and we will probably alternate between both of the speakers. I will start with Dr. Mendizabal, talking about this first live birth, which is actually a major milestone, but we would like to know, from your point of view, what are the remaining scientific and regulatory barriers that must be overcome before remotely operated ICSI becomes part of routine clinical practice? There are a lot of issues here to discuss, I guess.
Yeah. So of course, if we're gonna do remote ICSI, one of the first questions is who is gonna be the operator, right? Like, because it's not. We should be able to have a good way of selecting or given maybe license who is going to be able to be on the other side of the computer. Of course, training is going to be needed, and people have to prove that they are capable of working with that. But apart from that, of course, there is no this whole AI wave that we are seeing in not only in IVF but in many other fields. There are not that much regulations right now, but I'm sure they're going to come. People are going to get together and start saying, okay, which things can be driven by AIs. And of course, I'm sure there's going to be some requirements for these AIs to be used. So I don't think right now there's a limitation for that, not that we know. But for sure in the future, there's going to be something. So yeah, I will say that right now it's not that far that we could use these systems for as long as you have the system, right? And of course, there's going to be some requirements in terms of the speed of the internet and things like that. And the worst thing that can happen if you're doing remotely is that you lose the internet connection. But of course, there should be also some rules about who is going to be there in the system to take over, because you should not stop. Like, you have to continue with the procedure. So this is not that clear right now, but I'm sure in the next future we're going to have to answer all those questions.
Maybe this will have also implications in the informed consent.
Yes, of course.
Okay.
Yeah, that's true.
If I could just add to this discussion that, in my view, remote operation is not the point here. The point is automation. Automation makes it possible to perform this procedure remotely, but that is not the point. And I think that for automation of a complex procedure like ICSI to be implemented and to be accepted in IVF by IVF practitioners, it has to perform either better than manual procedures or at least equal to manual procedures, and offer other advantages, which the most important of which I think is consistency, which is difficult to maintain when you do these procedures manually. So I think the barriers, and of course regulatory barriers, could be real and numerous, although at the moment it's not clear really what those regulatory barriers are. I think the main barrier is performance. It has to do better. We can't take 10 minutes to inject an egg, right?
Really? Yes.
Thank you. It's a very interesting point, this automation. This is a very interesting question, going to continue with the same line of discussion from Bronte Stone from the US. I mean, you do this, or you design this automation system or robotic system through what you have learned in doing manual ICSI. So the question is, should humans incorporate anything that you have learned from this automated ICSI? Like,
Well, right now we're trying just to mimic what embryologists do, and that's by itself a challenge because humans are very good in controlling the. I think the main challenge is the aspiration part, right? Because the positioning of the needle and holding pipette is not a big deal, but the aspiration part, positioning the sperm inside the needle whenever you want it to be, and being able to release it on the cytoplasm, I think that's very challenging. That's what we're learning. So we're trying to imitate that from humans. But I think one of the things that we have also developed some other methods to pass through the zona pellucida, which we're going to present later, which I think will be valuable for the manual lab in turn. But we still need to do some research and prove that it's safe and all these things, right? But I will say not right now. Let's just try to be as good as Mina say, as good as embryologists to start.
So the one point, again, I'd like to add to this. If you examine ICSI very carefully and the various steps involved in it, the question is, have we actually optimized it? And there are many aspects of ICSI that have not been optimized and standardized. We are here in this system, the system is mimicking pretty much the concepts that we have adopted during manual ICSI. And the question here is it adopting these as optimized practices, or is it just adopting these as things that have been done routinely during manual ICSI and are being adopted as they are? The process of automation can help. It can help figure out what is the optimal approach. At the same time, the other side of the coin is, if you try to automate something that is not optimized, then the automation is going to show the same limitations that the manual procedure is basically facing. So, yeah, it's a very deep question. It's a very good question.
A lot of things to learn, for sure. Let's ask a question to Dale or Steve. There's a lot of questions, sorry for them. We understand that probably the main mechanism for improving the quality of the sperm is avoidance of centrifugation or induced damage. But I mean, many of these new sperm selection technologies demonstrate improvements in lab biomarkers, but not always in the outcome, in the live birth rate. What level of clinical evidence do you think should be required before biomimetic sperm selection becomes a standard practice?
Yeah, I think we're planning on doing a split sister oocyte study for the device. And I think, yeah, it's a good question. I mean, patient selection is a good one. We want to focus primarily as a start on patients who have high DFI. So if we can address that as a first goal to see if it equates to better outcomes. But I think the clinical testing needs to be patient focused and focus on the right population first. It might not be the solution for every single patient, and specific patients have sperm that can do pretty well in DGC, but I think focusing on those patients where the avoidance of centrifugation is going to be a big benefit to them. Yeah, it is a good question. There needs to be a lot more investigation into where it fits in.
Yeah, and obviously follow it through all the way to all of the outcomes that your clinic's already recording, right? So look at the FERT rate, look at the embryo development, live birth rate, and all those need to be recorded. Go beyond DNA fragmentation for sure.
This class.
There are quite a few questions about these the types of samples that can be processed. For example, Elisabetta Piatti is saying, can we use this device for samples with a very low percentage of motility? Can you for example, another one, Dorte Egberg from Copenhagen is asking, based on your presentation, total motile sperm is very low in the microfluidic group compared to DGC. Do you recommend this method for the preparation of ICSI samples only, essentially? Do you have a max total sperm input? Can the device be overwhelmed? So maybe you can define a little bit more in detail what the requirements are for the beginning sample.
Yeah, so because it's a passive device and it relies on the sperm's ability to swim through the device, we do have lower limits on what we would deem suitable for the device. Although you want sperm, if you have a population of non-progressive sperm, then we rather move to do a centrifugation-based selection. I mean, this paper was on quite an early prototype, so things have changed over time. But you want to have a population that you're able to harness the sperm's natural ability to traverse those channels at 37 degrees. So they'll become activated through the process, and you're able to isolate. But you don't want to make it difficult for the embryologist to be able to find sperm in the output. For the device at the moment, we do think that it's best for ICSI, but it doesn't mean that it can't produce enough sperm that you would have enough for IVF. It's just the versatility is a lot better with ICSI, where you only need one sperm per egg. So those lower reference or those lower limits on total motile sperm and then initial concentration are kind of changing as we develop the device. But we're hoping to just increase the yield as much as possible. But in that process, we also don't want to allow the sperm that we don't want in the output to get through. So we still want to be challenging that sample and just taking as many of those good sperm passively, allowing their motility to self-select them in the output.
Yeah.
So have you compared the DNA fragmentation rates between the samples from your device to samples obtained from SwimUp?
That was the previous study.
Yeah, not in this paper, but in a previous one we have as well. And that was on a slightly earlier version of the device. But we saw a similar trend in that study, that the DNA fragmentation was significantly lower in the device. But like Dale said, the device has moved on a few generations since then. So I think maybe we'll have to come back and do that again on the newer versions and medical grade plastics and all that sort of stuff.
Yeah, I think it's an interesting question because it really goes to the heart of the mechanism. What is the mechanism? You're saying self-selection, but based on what?
Thigmotaxis. So there's some quite good research done about 10 years ago now, where they've showed that sperm that follow boundaries tend to have lower DNA fragmentation than sperm that within the same kind of geometries just swim straight. So the ones that actually hit boundaries and follow them have lower DNA fragmentation. So that's the kind of mechanism that we exploiting in these devices. So when they get to those, it's similar to what embryologists do. A lot of embryologists do during ICSI, where the sperm is in the droplet and it hits the kind of meniscus of the droplet, and if it follows that meniscus, a lot of embryologists will pick those sperm because they're kind of tracking through that. The side of the droplet. So it's the same concept, and there was some good foundational research showing that those sperm actually have a lower DFI on average.
Yeah, how DNA fragmentation is related to motility is the question.
Yeah, yeah. I mean, sure.
Yeah.
Yeah.
There's a question here also quite interesting because even though it's addressed to one of you, but I think it can be answered by both of the panelists. I mean, they're asking from a human in the loop perspective. I mean, should the embryologist simply trust the device to either, deliver the best sperm or do the best fixing, or should there always be an opportunity for expert review and intervention? Because we know that machines sometimes they fail. I mean, look at simple blood tests. Sometimes there's a process that is stopped and we need to redo it again and load again the blood serum or whatever. So what is the right balance here between automation, which I think we all are looking forward to happen, and human expertise? Maybe Gerardo can start.
Yes, thank you, Juan. Um, I think we need the human to be in the loop. The whole idea of automation is not to replace the embryologists. It's actually to make their life easier and better because this is a tool, right? It's a tool that they can use to be faster and use their time in a better way. So I will say we do need to have the embryologists there. There's going to be edge cases where the automation might not be the right solution, and then we need a human to intervene. We have seen this in other industries as well, right? For example, uh, how many autonomous cars there are, right, in the streets right now, but still it is required for a person to be aware of what is happening and take control if necessary. So I say um, automation will not replace embryologists. It's the other way around. Automation is going to give them better tools to make their work better and eventually achieve better results, right? We all know that the current results of treatment are not as good as the patient would like to. So I think that's how we should look at the automation part.
Yeah, I'd agree with that. And I think you're always going to need contingency, right? You're always going to need oversight. And this comes back to the question around regulation as well, right? Even just for something as simple as our device, um, you have to look at the risk factors and how you mitigate those, and there has to be some sort of response, even if our device didn't work in a case and you didn't get sperm out, you have to be able to respond and work off that. But I also think it's when you're comparing for these edge cases or when you're comparing against risk factors where you need a human involved, it's important. Like no system is going to be infallible, right? So what's the baseline you're comparing to is humans doing these tasks, embryologists doing these tasks, right? And nobody's infallible. No one's perfect. So it's important to compare, I think, what your current baseline is, especially with something as complex as this automated ICSI system, is what's the baseline that you're comparing to against humans doing the process as well. Yeah.
There's another question also that probably is good for both of you. And I would like to hear your personal experience because it's a question about the patient perspective. And how do patients react when they are told that either fertilization or sperm selection was performed through a robotic system? What is the feedback from the patients?
So I think in our case is very positive. In fact, these are IRB studies that we are performing. We are doing simply no size and this, and the patients know that this is happening, but they enroll to this study knowing that half of their eggs will be processed through the automation. And I think overall they are very positive. They really believe in that this is possible. So I think overall, in with respect to patient, is very positive feedback, I will say.
Yeah, even similar to us, like with our technologies, although they're far simpler, the response has been very positive, and recruitment hasn't really been, an issue. Consenting patients has been quite smooth, actually. I think they're excited about innovation. Patients, especially male factor infertility, they're quite driven and they're looking for solutions and they're quite well educated, actually, about their treatment and what the options are. So we get a lot of patients reach out to us asking about the studies that we're running and they want to be involved. So also the same with our AI from the previous year. Similar story.
I think our task is to define risks and benefits, and these have to be very well defined for the patients. And obviously, that's the purpose of an informed consent. It is one thing to participate in a study. It's another thing to say, well, this lab is completely automated, and when you enter this lab, everything is going to be handled by machines and robotic systems. So then it becomes a question of, okay, so what am I risking? What are my risks? And so I think it falls onto the providers of these services to define those risks well enough for patients to be able to make informed decisions. At the moment, that's difficult. Even with AI, we have not developed very solid frameworks and definitions of risks, and that still remains to be done.
Yeah, I definitely agree with that. We have to be able to communicate, we have to be able to understand. And it comes back to what we're talking about, the human in the loop factor, the regulation, how do you mitigate those risks? And it's an ongoing process. It never ends, right? It doesn't even stop it being regulated. You have to monitor and have post-market surveillance and keep yourself up to date. Yeah.
All right. So I think being conscious about time, I think I would like to thank you all who joined today. My sincere thanks to my co-host, Dr. Mina Alikani, and our speakers today, Dr. Steven Vasilescu and Dale Goss and Gerardo Mendizabal. It's been an absolute pleasure to talk to you today and have you with us.
Thank you so much, Juan, and thank you, everybody, for your presentations. I would like to also recognize the I3 organizing committee, Jacques Cohen, Peter Nash, Marianne Sabatis, Thomas Elliott, Fran Farley, Violetta Sora, and of course, Giles Palmer. We will be back with another RBMO webinar in September, but coming up next week from I3 is New Rules, a webinar devoted to innovations in PGT and sperm preparation. As we talk today, there is so much interest in this topic, a lot of interest. So that will be on Tuesday, the 16th of June, 2026. Don't miss it. Thank you again. It's been a pleasure, and see you soon.
Thank you. Bye-bye.
Bye-bye.